Today we're going to be talking about transcription in prokaryotes. There are four main steps in the process of transcription. And the main purpose of transcription is it's just another pathway to be able to express genes. I'm going to be using pipe cleaners and other craft supplies so that you can easily follow along if you would like. First, we're going to start with template bonding and initiation. Here's a strand of DNA. RNA polymerase and holoenzyme put together the promoter region, which is just where the DNA sequence binds to initiate transcription. The core enzyme and sigma also bond to this promoter region, and the DNA double helix starts to unwind. Let's zoom in on this segment. The +1 at the bottom denotes where transcription started. Nucleotides begin to be added to the 3' end to synthesize mRNA. The NTPs help with this RNA synthesis. Only one strand of DNA is transcribed, and that's called the template strand. The mRNA product is complementary and almost the same. The coding strand is the second strand. Notice how the RNA strand is 5' to 3' and the template strand is 3' to 5' because they're antiparallel. DNA is still being unwound and then rewound by core enzymes. During this time, the sigma factor is also released. Now that DNA has been transcribed, it must now release the RNA product. There are two modes to go about this. The first is row independent, and there are no external proteins needed, so it's just RNA based, and that consists of inverted repeats and then about eight TA pairs. And when it transcribes, hairpin forms, which is what you see right here, and it's a loop, and base pairs are pairing with themselves to create this loop. So when it's transcribed, these are UA pairs, and they're very weak, and so the RNA detaches. The second is row dependent, and it depends on this row protein, and so there's C on the RNA outside, and the protein binds, and ATP transports it, and it unwinds the RNA so it detaches. Now we're going to talk about the -35 and -10 regions on DNA that the sigma factor binds to. These regions just have similar sequences that are found in many prokaryotes. This -10 region is called the Tata box because of its TA-TA sequence. I hypothesized that if this box was deleted, that because it determines where the genetic sequence is read, that the enzyme would not even be transcribed, and therefore that organism would die because it didn't have that enzyme.